I'm very excited to be here presenting for my first minimal cell workshop. And I guess you get a feeling from previous talk by Natasha and Isaac. But basically what our lab really wants to play with is the dynamic between you have different lipid structure, which is different chemical structure, and how that affect lipid-lipid interaction. So membrane biophysical property, and then directly how it affects the cell, so a biostructure. And through this talk, I hope that I can convince you that Mesoplasma is a wonderful organism to be working with for this type of question. And without further ado, let's start. So I think we know that cell membranes are lipid membrane, and they are composed of mostly phospholipid that looks something like this. With, you can easily separate it to one more hydrophilic head group, and the long chain hydrophobic. And the cool thing about lipids is that lipid chemistry actually changes the membrane biophysical property. For example, if you have a lipid with only saturated chain like this, it would have a high melting temperature And if you introduce a double bond in the middle, then the lipid melting temperature would be lower. And what does that actually mean? It means that in normal condition, in room temperature, high melting temperature lipid can form a gel phase. This will restrict the lipid diffusivity, therefore, multiple enzymatic process cannot take place as they would like to. But if you have a low melting temperature lipid, the lipid would be in liquid disorder state, and then the membrane might not be as robust towards outside forces. So that's why in eukaryotic membrane, there is another type of compound that is very important, which are sterols. So what it does, it is create a liquid ordered phase that is something in the middle, that you still have diffusivity, but the membrane is in a more ordered state. And I think everyone is familiar with a very common sterol, which is cholesterol. So basically for eukaryotic membranes, these are the three main components that is like a must-have. You need sterol, you need low melting temperature lipid, you need high melting temperature lipid. However, we also have bacteria as well, right? And bacterial lipids are different. So most bacteria does not produce sterol at all. And some of them rely on a family of compound called hopanoids, which are considered sterol analog. So the cool thing is, in 2020, we actually tried to look into the lipidomics of this bacteria, Methylobacterium extorquens, which have hopanoid. And not only does it have hopanoid instead of sterol, like in eukaryotes, the lipid unsaturation site is also different. While most eukaryote has unsaturation site at the delta 9 position, our bug actually have the main one at delta 11. And what is even more fascinating is when it's adapting to cold temperature, they actually produce another double bond in the delta 5 position, which sparks our interest. So why exactly are they doing this? How does moving the double bond affect sterol or hopanoid condensing effect? Or in general, how does bacterial lipid interact with one another? So to delve into this, I first take a few biophysical approach in an in vitro system. And the system I chose is the monolayer. Basically, what you want to do is you put two lipid on the monolayer. If you put them separately, they have the area of, for example, A1 and A2. But if you mix them together and they yield a smaller mixed area comparing to the sum, then it's called condensation effect. And this is a diagnostical effect of cholesterol or some other sterol as well. And we want to see how does it work if we add diplopterol or hopanoid to the mix. And this is the result. Basically, you can see that cholesterol can show condensing effect to lipids with different double bond position, all the same, maybe with slight differences. But for diplopterol or hopanoid, that's not the same case at all. Especially if you look at the delta 9 and delta 11, you are literally just moving the double bond 2 carbons away, and you yield a completely different phenotype from condensing, or which means the 2 lipid has to pack very efficiently together, or for the or negative condensation effect, which is the lipid might be more repulsive. To gain a deeper understanding into this, we collaborate with Ed and Liam from the University of Delaware to look into how the different methyl group distribute according to the double bond. And what we find very fascinating here is that for the delta 9, which have a repulsive effect, the methyl group of diplopterol is actually distributing exactly where the double bond is. And there might be a steric hindrance that stops them from actually packed closer to one another. This did not happen for delta 11, which might explain why they can pack together and have create this condensing effect. So far we see that there is a link between chemical structure towards biophysical property, but how does that link to any biological organism? So this is where Mesoplasma come in. Oh, I have the citation here wrong. Actually, this picture is from Sebastian Rodriguez group, who has 3 talks that will be presenting right after me. Sorry in advance. So this is Mesoplasma florum. It's a molecule like Mycoplasma, so it only have 1 plasma membrane. It also scavenges lipid from the media, so that's how you can control and create a membrane defined lipid membrane. So what I did was adding different lipids to this bacteria, some saturated lipid, the unsaturated lipid accordingly to the 2 different ones that I found. But what is so special about Mesoplasma florum is that it does not rely on sterol. So basically I can choose to add either cholesterol or diplopterol, a hopanoid, and see how different lipids interact with one another here. As Isaac also introduced this, we want to test the membrane robustness through an osmotic shock assay, when basically you apply hypoosmotic shock to see cell popping, and you stain it with propydium iodine. It's a DNA stain to see how much of the cell membrane that has been compromised. And here you can see that if you feed the cell with cholesterol, which is the one on the 3 bars on the left, there is barely any differences if you feed them along with delta 9 or delta 11, but the changes are way more significant if you have delta 11 and diplopterol comparing to diplopterol and delta 9. So basically what we gain from here is Mesoplasma as an organism has shown that if you feed it different lipid, you can control the membrane robustness and its sensitivity to osmotic shock. But if we flip the switch, we also see that Mesoplasma has a rule of its own. So basically, what we try to do is we feed the bacteria Mesoplasma florum and Mycoplasma mycoides the same lipid diet, which is undefined one, FBS serum. And we look into the lipidomics of these 2 organisms. And as you can see here, even per lipid head group, which are a pretty generic feature, you can see that they are using different lipid. While Mesoplasma florum use more PG and DAC, Mycoplasma is using way more PC, sphingomyelin, and cardiolipin. Well, what does this actually suggest? It suggests that these 2 organisms, even though they are relatively closely related, their different genetic background and settings are forcing them to have completely different lipid membrane. And through Mesoplasma, we can understand a different way that lipid membrane can remodel, even though we are applying the same stresses towards them, for example, temperature or antibacterial. So this is what I would want to do in the future. So the take-home message here that I want to present to you is that first, biomembrane property can be modulated through its lipid. The second one is Mesoplasma is great to explore all the bacterial lipid combination because it does not require sterol in this diet. And also Mesoplasma, although small and relatively similar to other mycoplasma, I have presented a new platform for you to understand lipid remodeling for temperature. In the future work, I would want to focus on the Mesoplasma remodeling on defined lipid diet, and also the evolution diversion between hopanoids and sterol, which are the two lipids that I'm very interested in. So that's the end of my talk. I think I talk way too fast, but thank you all for listening. And I would love to address any question that you still have.
Questions for An? Anyone? So my thought is, as you have the mycoides, so mesoplasmas and mycoplasma mycoides are very similar, given that we can transplant a mesoplasma genome into a capricolum and get a cell, and we can transplant a mycoplasma mycoides into a capricolum genome and get a cell. So they are very, very similar, but the lipids are quite different. How much power does this give you to investigate lipid properties with the two systems relative to one?
I mean, lipid chemical structure definitely have to play a role in the biomembrane properties, right? But if you ever look into the lipidomics of different organisms, you can see that they have basically all types of solution. You have sterols, you have hopanoids, you have lateranes. Some bacteria maybe use less PC comparing to eukaryotes. They have all types of different solution. But what are the problems that poses to them? They're relatively the same, or don't you think? That they have to adapt to a certain temperature, they have to be robust enough to a certain extent. So basically, what I see when I look at Mesoplasma comparing to mycoides is that you have two machinery that are trying to adapt to the same thing. And here you can scan how different combination of lipids can actually achieve this. Because if you just go into mycoplasma, you are limited by the mycoplasma genetics and enzyme machinery. While if you go with Mesoplasma, they are similar, but from my data, you can see that the lipidomics makeup is completely different, right? So I guess it's like we are trying to understand how multiple lipid structure in combination to one another can yield a relative can provide the same scaffold for cellular growth.
I think about old experiments where Shlomo Ratem was pushing Mycoplasma capricolum or mycoides to grow without cholesterol. So I'm thinking, you've got your two systems. Could you gradually adapt them to live on each other's sets of lipids?
I think from my personal experience, I try to dilute out all the cholesterol and replace it with diplopterol in mycoides. Mm-hmm. And I still, at the end, I still need a little bit of cholesterol there. So it might be playing a role as a signaling molecule, for example, or some protein needed it specifically to work. So this is what I mentioned by you are limited by the genetics and the enzyme that mycoplasma have. So
will Mesoplasma grow on SP4?
Actually, right now I'm using SP4.
Okay. with fetal bovine serum?
No. So I have both of them, both on fetal bovine serum and also a defined lipid diet.
Okay. All right.